My name is Gabriel Pereira. Bronte, did you want to introduce yourself?
Hi, my name is Bronte Lucia. I'm the Education Manager and Technical Sales Manager here at Aura Health.
Dr. Benny, enough about us. Please, if you could give us a little bit of background on your background and particularly your background as it relates to the Turmacin study.
So my name is Benny Antony. So I'm one of the senior research fellows at the Menzies Institute for Medical Research, which is part of the University of Tasmania. And I'm also a manager for one of the primary health networks here in Tasmania, Primary Health Tasmania. So currently for this clinical trial, I've been doing clinical trials as my passion, and I'm mainly focusing on osteoarthritis or other musculoskeletal diseases. And then I've got a special interest in the natural remedies, and that's how I came up with this Turmacin study.
So go back a step. So that's fascinating. So what's your interest in musculoskeletal disease? Where does that come from?
Yeah, so I did medicine back in overseas, and then I moved over here. So I did a PhD related to musculoskeletal diseases because I was really getting into initially planning into research in orthopedics or practicing in orthopedics. But and then once I came over here, I did my PhD related to musculoskeletal diseases, particularly osteoarthritis. And then we introduced some new biomarkers like MRIs and all those things. And then we started implementing those MRI objective measures in clinical trials, and then making our clinical trials much more objective rather than just measuring the pain.
Objective evaluation. Yeah, exactly. And why natural products? What we're delighted you're interested in natural products, but what sparked your interest in natural products?
Well, obviously, the main thing about natural products is that there is not good quality, clinical trials available for osteoarthritis, although the popularity of these natural products is quite high. So I am a supporter of natural products, but at the same time I'm a bit concerned about the toxic effects of these chemicals, which just because it's natural doesn't mean it's safe. So I always had that point. So I wanted to really investigate these products in a scientific way, in the most rigorous rigorous methodology, using the rigorous methodology. So that's how I came to into this field. And obviously I have an Indian background, so yes. I came from I don't know whether turmeric. The best content of curcumin is in the Alapi variety of turmeric. So I come from the region which is close to Alapi. So
forgive my ignorance, where is that in India?
So that's in Kerala.
Oh, amazing. Okay.
Yeah, so you can nearly, obviously that the content of curcuminoids is quite low in turmeric, and I think the Alapi variety maximum can 6 to 8%, so which is the topmost variety. That's why those kind of turmerics looks more brown. And then obviously as you go like South Indian, other Chennai variety, they look more like yellowish.
Yep.
So probably the brown content is more, maybe indicating more. I'm not a chemist, but that's my understanding.
But same species, yeah?
Yeah, it's all curcum longa, but the content of the curcumin content is different.
Fantastic. So Kerala is my my Ayurvedic spiritual heartland. It's basically where I discovered Ayurveda for the first time. So I spent postgraduate, I spent 9 months traveling around India and spent probably the majority of that time in Kerala. I fell in love with the place. It's amazing.
It is a beautiful space. I had a good time there as well.
And the food is just incredible.
So
because
good.
I think it's like Sri Lanka, right? So there's still that Portuguese influence flowing through as well. So just the composite culture and food is just breathtaking.
Yeah.
Amazing.
Thanks. Yeah,
anyway, we should stop talking about travel.
That's right.
So but that's a great segue, because I think, look, my view is this: the best researchers in natural medicine are the most pragmatic and skeptical, right? So you have people like Bronte and I who are natural medicine tragics. We have an underlying belief in tradition of use, in kind of anecdotal data. But as we were saying before we began recording, really the gold standard is that double-blind, placebo-controlled, pharmaceutical standard clinical trial structure. And I think if I'm hearing you correctly, that's exactly what you're looking to bring to your practice of research, but in particular, this most recent study on Turmacin.
Yeah. So yeah, so when it came to, when we decided to conduct a clinical trial in turmeric extracts, so the first thing which I was curious was to see what is there and available in the literature which might have the better, therapeutic potential. So the first thing is to identify an agent which is, which might have a better benefit to human human osteoarthritis patients. So that's how I was looking, and there are plenty of extracts: natural extract, bioenhanced extracts, semi-synthetic extract. I believe you probably know the flexifenol as well. So that's a semi-synthetic extract as well. So there's plenty of options, so conjugated options and, like piperine mixed options. So you guys are the experts in that. But
yes.
So when I was going through all this literature, so it may be hard to quantify the effect of any of these because all the trials or all the studies use different methodologies and you cannot really compare each one.
100%.
So some of the trials.
There's no Cochrane or equivalent meta-analysis on turmeric, of course.
I mean, we have done a meta-analysis on it, but it's impossible. Okay. Yeah, we have. So if you look at, I'll send you those papers. We've done a meta-analysis on that, but we've rated all the quality of the studies quite low because some of the standardized mean difference of some of the studies, especially one conducted in India. I think it's used Turmacin as well, but and one in Iran and all standardized mean difference of 3, which is impossible. I'll say that, which is like, if that was true, then none of the doctors needed to be there, exist. You give the medicine and that's gone. The pain is gone.
So I want to get you to comment on this because this is something that I think a lot of our audience do not understand, right? Yeah. 2 things. One is power calculation and statistical significance in clinical research. So I'd love to get your perspective on this because it's a really it's a hard thing to achieve, right? So and the importance of study design cannot be overstated.
Yeah.
And then particularly in studies like osteoarthritis studies where there's a significant placebo effect, how do you account for that, up to 60, 65, 70% placebo effect?
Yeah, you got it, Gabriel. Most of the people doesn't understand the placebo effect. So that's a very good point which you made, like the 60%, 70% of any effect of any treatment in osteoarthritis, just placebo effect.
Yep.
So, but the problem is that if you look at the previous clinical trials, most of the clinical trials in turmeric, you probably literally see almost 0 placebo effect. That means their blinding wasn't perfect. Although they say that for systematic reviews, when we assess the quality of the studies, if you have mentioned that they have double blind or then we have to put that tick. So they might meet the criteria for fitting the systematic review as a high quality study, but we can see from the trial, when we really know the game, we know that it's not properly done because it's impossible to have, such a, almost 0 or no placebo effect, so that's why I'm saying, it may be hard in a systematic review to criticize it in a methodological way, but I always in my systematic review put a paragraph of my opinion that this is almost impossible.
And sorry, I don't mean to rabbit on, but this is one of the considerations around sample size, right? So study sample size, because this placebo effect is so large, to achieve statistical significance, you actually need a population study sample of like 300 plus people, right? In some cases.
Very true, very true. Because so the sample size heavily depends on, obviously, the big difference between the expected difference between the placebo group and the treatment group. So, if you expect a placebo response of 0, you need only a very small sample size because, you're going to get a big difference, right? But when we are estimating a sample size, effect size of smaller difference, of 10 millimeter. In our case, we powered our study for 18 millimeter. But I'll get into all those details later. But as I was mentioning, the power sample size calculation depend on the and the difference between the treatment and the placebo group you want to estimate, and the standard deviation of the variable, which you're observing as well. So a lot of the studies are smaller sample size. Just because it's smaller sample size doesn't mean it's a bad thing, because our study was a smaller sample size study as well. So this was designed as a pilot clinical trial, which we wanted to take it to the next stage as a proper phase 3 clinical trial, and which we are still seeking funding.
Let's double click. Sorry, it's turning into a podcast. Let's double click on that. So generally the approach is in vitro, in vivo animal models where that's appropriate, phase 1, phase 2, phase 3, as you say. So this is a phase 2 study.
Well, we designed
phase
it
1, phase 2.
because since it's a natural product, it doesn't have to be listed as a phase 2 trial because, if it is a phase 2, usually people divide it into phase 2A and phase 2B.
Yes.
We can probably maybe depict our trial as to more into phase 2B. Phase 2B trial. Phase 2A is more into, again, more than efficacy. It will be more into safety again, phase 2A as well. So phase 2B is particularly interested in the efficacy point as well. But anyway, so our trial was, it's not a fully well-powered sample to detect the meaningful effect. But what we found was, we found an effect of 9 millimeter. We estimated an effect size of in 100 millimeter scale, VAS scale, we expected a difference between the group of 18 millimeter. So that's what we powered for.
Because our power for the audience at home, visual analog scale.
Analog scale. So, but we observed a difference of actually 9 millimeter, which is half of what we estimated as the minimum clinically important difference. We saw half the difference. We still found it statistically significant because the standard deviation was half of what we expected.
Yeah.
So it compensated. That's why we could find a statistically significant result. But that means that but the effect size was only 9 millimeter, which means that we did not find that just by giving a turmeric capsule, your pain is not going to go like massive reduction. It's a modest effect.
Yes.
That's why this trial is more important. And if you show this trial to any clinicians or anybody, they're more likely to believe our trial than any other trial which existed in turmeric, which is showing massive effect, so you take this medicine, some of those trials which I don't know, if you look in the 49 millimeter difference, which is impossible. I don't know whether nerve growth factor, NGF blockers, tanezumab trial. So it's like a nerve growth factor, it's like a monoclonal antibody.
It's a pain block, basically.
Yeah, and even those trials in osteoarthritis that hardly found 0.5 was the standardized mean difference, whereas this turmeric trials are finding an effect size more than 1 or 2. So it is.
In addition to impacts on, say, WOMAC scale, VAS scale, reduction of pain, symptom kind of amelioration as well, you're also with natural product trials, and I'm sure you did as well, looking at side effect profile.
Yeah,
so I can be on silazepam, I can be on ibuprofen, and the impact long term is not great for my gut. It's not great for my kidneys. It's not great for my liver, right? I might get pain relief for a period of time, but I'm not addressing anything beyond symptomatic kind of relief, and I'm also having a side effect profile that maybe is not very desirable. So I think, and this is something you guys covered in the trial.
Yeah. So every clinical trial, which has to be really good quality, because our trial was published in Annals of Internal Medicine.
It's a great journal.
It's the topmost internal medicine journal. So our journal was published mainly because we ticked all the boxes, including collection of a proper, adverse event data. So adverse events have to be captured even if it is not related to the treatment. For example, we'll capture anything that patient reports, they have something. So it doesn't have to be related to the treatment. And then it goes to me like the principal investigator, and we assign whether causation or not, because we are blinded as well. So in our trial, we did not find any. There were maybe, and I don't remember exactly how much was, there were actually 24 adverse events, but they were both equally distributed between the treatment and the placebo, which means that, there was no significant increase in, with the treatment, there was nothing we can assign as an adverse event for this particular trial. But again, you have to remember that we used a dosage of 1,000 milligrams. So which included 85% Turmacin, which is a polysaccharide-rich extract of turmeric, and then we used a 15% curcuminoids as well, which is an oil-based. I believe it's BCF90. I don't know. Maybe if you ask. So it's like an turmeric oil-based extract. So because we came up with this combination because, as I was telling, I specifically asked actually, that time I forgot what was the name, Dr. Your scientist in the Dr. Parthi might know. Who was your scientist's name? I forgot.
Dr. Bharti?
Yeah, but it's okay. Doctor, who was it?
On natural remedy side?
Natural remedies. Yeah, who was your scientist's name? Was that Doctor?
Is it?
Or Dr. Deepak?
Sorry, yeah, Dr. Deepak. Yeah. So at
Dr.
that time,
Incredible
when we
guy,
were
like one of the greatest phytochemists that I've come across in terms of Ayurvedic extracts. He's fantastic.
Yeah, so we were discussing this that time actually, and then so I particularly said, like, okay, so you found that the PCF90 has got effect, and then Turmacin have got effect, so why don't you combine? So that's that always tell, like, so because I'm looking for the best extract,
Yeah,
I know that we can still combine with Boswellia and all those things because Boswellia, but I wanted to make it like a single trial, because there are trials that showing the turmeric whole extract is also like from, I think it's from I don't know where, somewhere Southeast Asian countries. There are a couple of trials which showed that the whole extract was effective as well. So this somewhat mimics the whole extract of the turmeric. So the combination of BCF90 and Turmacin. So
so we loved it from a commercialization point of view because, as you say, it's almost like what we call a full spectrum extract.
Yeah, and that's when they were telling, like, they were planning to make it as a commercial extract to name it as Turmacin Plus or something. And I don't know why you guys
Turmafort, I think they were going to call it.
Okay, I don't know why it was dropped, and I don't know the commercial side because this was an investigator-initiated trial, which I did,
cool.
So this is nothing to do, but they did help me with the support financially. Got support, but again, it was an investigator-initiated trial. We got the support from them.
Yeah, so I think it's again worth double-clicking on that. That basically means, as I understand it, you reached out to Natural Remedies and said, "Hey, I want to do some research on turmeric, on Turmacin. Will you please supply me some material to do my trial?"
Yeah, and also they supported me financially for the MRI component as well.
Got it.
So it's not in any way design. They didn't have any contribution in terms of the access to data or design of the trial or anything. So
it's your study.
Yeah, it's a study. So that's what we say, the investigator-initiated trials. And I'm really thankful to Natural Remedies for that, because I've already mentioned it everywhere in acknowledgements and everything.
I love that, and I'll tell you why. Because it is risky as someone who sells an extract to have someone totally independent do research on it, right? Because whether it's good or bad, you have the right to publish.
Yeah, that's right. Because they didn't have access to the data as well.
Right.
But and same time, they were even kind enough to contribute a bit more to my biomarker analysis, which I complemented. Again, their support is not enough. I just put my fellowship money as well, and recently published some biomarker analysis. Unfortunately, there's no significant difference. That paper is also just published 1 month before. So that's from the same study. So
but
because
it's the purest form of research, in my opinion, because you've reached out to them. They can't control the publication outcome. And I think it's commendable for you and for Natural Remedies to do this sort of research. Because you have to believe, the point I'm making is you have to believe in the extract. Yeah,
that's right. So I reached out to them because I knew that component, and then I also knew one person who worked there. So I discussed with one of my friends, and I worked, and he was the one who introduced me to Dr. Deepak, and that's when I started that I wanted this combination. And then he said they were planning something that time. I don't know. I think and you were doing some basic science research that time in animal models, and he showed me some data on this Turmacin. Right. At that time, the name was Turmacin Plus, right? I think that's my understanding that time. So they said they had planned to do that and commercialize. I don't know what happened to those things. And then that's how we fixed on it. He wanted it slightly more. I wanted 20%. I think he then moved on to 15%. That's how, we had a lot of discussion, and that's how that percentage was agreed. And then and then we decided to go with the trial, and it was a because conducting a trial with a drug which is supplied from overseas was a big challenge for me, because I tried to conduct a similar trial the second time, and because it was conducted a few years back, it went through with the TG and all, and then the second time when I was trying to do it, didn't go ahead. So I think things have become much more stricter now. So it's not that easy anymore to do those kind of trials anymore.
No,
So
no.
anyway, so I was lucky enough to get all those approvals and everything, and then conducted the trial here. And then, so as I mentioned, there was a statistically significant, modest effect of the drug or the the turmeric extract for knee osteoarthritis.
So I think this is fascinating for our audience anyway. So you've designed your study, you've got ethics approval, you've got the approval from the regulator.
Yeah.
You can then begin recruitment. Who are you recruiting, and how do you do that? And are they seen in a hospital setting? Are they seen in a clinic setting? What's the program?
Okay. So first stage was, obviously I had the proposal, and then I secured some internal funding from the hospital itself. So we had, I told you, like, this was an investigator-initiated trial. So the majority of the funding came from internally from the University of Tasmania and the hospital. So we got the funding, and then the other half we got it from the natural remedies as well. So, and then with that funding, obviously we need all those ethics and regulatory approval, including the clinical trial exemption, so from the TGA. So we got all the approval, got the drug,
So
and
the
then
CT
exemption. So,
so
sorry, no, this is not exemption. This is, sorry, because so many trials. The current one which I doing was the CTX. This is CTN. So that means clinical trial notification. So we still need to notify them, but but right now with the
So this is
because
just for the people listening eventually. This is a regulatory exemption with the regulator, with the Therapeutic Goods Administration, which allows people to study natural compounds where safety and toxicology can be established. So no toxic effect, safety effect is there, but it doesn't need to be manufactured in a TGA licensed facility. Is that correct?
Doesn't need to be TGA. It doesn't, but it still need to have to be, the GMP certified facility and all those things.
But not TGA, I think. But also you can use it for what we would call off-label potentially indications.
Yeah, because I think the Turmacin was already there in the ARTG, Australian Register of Therapeutic
Goods. So that would have helped a lot.
That was the main thing which helped. Otherwise, I couldn't have imported the drug, got the permission to do it. So anyway, so a lot of, because we had to submit a lot of papers and documentation and all those things. Anyway, I got the approval, and which I say again, I don't know whether if I redo the trial again, it would go through, because every time things change, every year things change. So and the drug, we recruited 70 knee osteoarthritis patients in the clinic at the Menzies Research Institute. So 70 knee osteoarthritis patients, which have got clinical knee OA, so which will be examined as a clinical knee OA, and they need to have a particular amount of pain. We say that 4 out of 10, so moderate amount of pain. And then we also desired, at that time, this trial was designed specifically because I wanted to see if there was an effect for people with more effusion for this. Effusion means, a local evidence of inflammation in the joint. Yep. So the hypothesis that time when I designed it was that the people with inflammation in the joint might reflect more an inflammatory phenotype of osteoarthritis patients. Yep. And the basic science evidence shows that the Turmacin or the polysaccharide rich component and the BCF9, the curcuminoid rich component, seems to reduce inflammation. Yes. So the idea is that we select an inflammatory phenotype of osteoarthritis patients, and then we supplement them with this turmeric extract, and then they seems to reduce. Then they will reduce the inflammation and thereby reduce the pain. Yep. So that is the basic
just
hypothesis.
In terms of design, typically you'll have a rescue medicine in there as well, so paracetamol or something equivalent.
Paracetamol.
And then that I think it was 70 subjects. They're split into 2 groups. One is the active, one is the control, right? Or one is the placebo.
Yes. Yeah, one is the placebo, so one is the active group, so which is just like any normal randomized clinical trial. So they were randomly assigned. It's a double blind, so neither me or anybody who is conducting the trial know which drug which they were doing. We were asked to, the drug was matched even for, even if the participant opened the capsules, I asked them to, it has to look the same, because I told them to color it yellow because people might open it up. So everything was done. So we tried to make it like a double blind, well matched placebo controlled clinical trial. And then the participants went on for the treatment for 12 weeks. So at the baseline, and when they were recruited in the clinic, I did their ultrasound scans as well to select people with inflammation. So that means the local joint of evidence of knee swelling. So we selected people with swelling as well, swelling, and then they were supplemented with the drug or the placebo, and then at the baseline, they underwent an MRI scan as well as all those.
Oh, wow, that's fantastic.
Yeah, so they did MRI scans at the baseline, and then they went all those pain, like the knee VAS, visual analog scale, and the WOMAC scale, and the quality of life, all those, the standard questionnaires which we use in a clinical trial. So, and then these participants were followed up over 12 weeks. So and during the time, obviously in between we used to get 4 and 8 weeks. We also got some pain measurements, but they came back.
What biomarkers were you testing during that time? Was it interleukin-1, or what inflammatory markers were you looking at?
Yeah, so at 24, at baseline and at 12 weeks, we collected MRI as well as the blood samples. Yep. And also at the follow-up at 12 weeks, we collected MRI and blood samples. So we had the biomarkers from MRI and the bloods only at baseline and 12 weeks. Yeah. So we looked at the changes in the inflammatory markers such as the CRP, IL-6, TNF-alpha, and then we also, in addition to what I spent some money into the biomarkers as well from my side, apart from their contribution, and then we measured the cartilage oligometrics protein in the comb,
which I loved as an element of this study, because I think there was some clinical effect in that regard, right?
Unfortunately, not in our study, but
in one of the previous Turmacin studies, I think.
Previous studies, because if I'll forward you my systematic review, which we did, and then in that systematic review, we can see some modest effect of the treatment from the previous clinical trials on these cartilage markers as well. They're not inflammatory markers; they are the metrics in, cartilage oligometrics protein, and then matrix metalloinoside protease as well, so MMP3. So they are cartilage specific markers, and they seems to have a beneficial effect in previous studies, but not in our studies. And we did not find a significant effect on the inflammatory markers as well. We did find an effect on pain, but that wasn't mediated by a change in the inflammation level in the systemic. But because our studies, again, I'm not saying 70 isn't that, a big study, So ideally, we do conduct clinical trials of 260 minimum here.
Yes.
So that's what our standard. So this was designed more or less, because with the limited resources we have, this was not funded by, and usually our clinical trials are supported by NHMRC, the National Health Medical Research Council. But this one was supported by, I told you, from the university and hospital, and then partially from the natural remedy. So this was a smaller budget trial, so that's why we had to cut down on the numbers and all those things. But again, there was no compromise on the quality of the trial. That's why we could publish in a really good journal.
In terms of clinical outcomes, so you've got a number of slides, I think, which I'd love for you to take us through if you like. But
I can share. There you go. Let's see. Yep. So can you see it?
Yeah, thank you.
Okay. So I'm not going to go through all those things. Obviously, I think people already know
covered a lot of it already, I think.
Yeah. So obviously, I'm not going to talk about what osteoarthritis is. Obviously, that there is no disease-modifying treatments for osteoarthritis, and it's a complex disease. So people initially used to think that it is no inflammation involved, and that's why people hesitate to call it as osteoarthritis. And previously, people used to call it as osteoarthrosis to avoid the itis, the inflammation compound. But recent evidence suggests that the systemic level inflammation, such as the CRP and TNF-alpha and IL-6, are associated with the progression of osteoarthritis. This is MRI evidence of inflammation. So this is what I was measuring, I told, as I mentioned. So this is a fusion synovitis, which is visualized in MRI. And this is in ultrasound. So this is exactly from a Curcuma trial patient, which we assessed. In ultrasound, I'll be able to see that effusion, this white bit will be in ultrasound, will be dark bit, because this is what I can see as the joint fluid. And we did not quantify for the synovitis because it's very difficult to quantify synovitis in osteoarthritis patient. But I wasn't, let's say it was difficult for me. But if you're an expert, maybe an expert in ultrasound, they would have been better able to identify it. But that doesn't matter because effusion was accepted as a standard criteria to define inflammatory osteoarthritis. So as I was mentioning before, there are previous trials showing, this is I think this Madhu 2013, trial is actually a Turmacin as well. So maybe if you were interested, you can do that trial. But this was a 3-arm trial. So I think this is from my memory, since I haven't given a talk on this for a long time, but this is a 3-arm trial. They did have a chondroitin arm as well, not just, so they had what I remember is they had 2 doses of Turmacin, and then one arm of chondroitin, and then one arm of placebo, I think. Anyway, the trial they found that there was a significant effect. Well, in fact, too much difference. I wouldn't, 5.63. Anyway, and these are some of the other trials. And I know personally this trial, Srivastava 2016. I personally visited this group as well because this is in Lucknow. So I know the person who did it, and then we tried to apply a grant with them called Australia-India Strategic Research Fund. We tried to do a collaborative research on this, but unfortunately it wasn't successful. So those people also conducted the trial, and then they found that there was a significant effect. In fact, this is the standardized mean difference. What I was telling, they are quite huge, but that's probably because of the design problems in the trial. But all of them say that there is an effect of osteoarthritis. So we wanted to take it to the next level, and then hypothesize that, as I mentioned, in an inflammatory phenotype of osteoarthritis patients, there will be a difference of 18 millimeter compared to placebo over 12 weeks. Out of 100 millimeter scale, we expected a difference of 18 millimeter. And, we, as I mentioned, we used an 80% aqueous-based extract standardized to Turmerosaccharides and a 20% curcuminoid fraction as well. And then we gave it to the participants 2 to 5 milligram, 500 milligram capsules per day as well for 12 weeks. Okay, this is what we we selected knee osteoarthritis patient above 40 years of age and with a pain of around 1400, which is same as 4 and 10, and they all meet the American College of criteria, and then also the ultrasound defined effusion synovitis. So this is the measurement schedule. As I was mentioning, at the screening we had all those vast measurement, also X-ray and ultrasound. And then at the baseline,
would they come into the clinic, or would they be having these studies at home?
They come into the clinic because they need to undergo the ACR criteria. So I need to examine their knee to make sure they have got the knee osteoarthritis. So that's based on the ACR, American College of Rheumatology criteria, because all the participants have to meet that criteria to enter into the study. Also, they need to enter the pain and other criteria as well. And then and we at the baseline they come again to the clinic, and they were given the capsules, and then we also measured their knee pain, and then also the MRI and the blood biomarkers as well at this time. And then they will be followed up over 12 weeks, and this is the time when they come though. The week 1, week 4, and week 8 they completed their questionnaires online at home. They don't come to the clinic, but week 12 they will come back to the clinic, and then they undergo again the bloods and the pain measurements and the MRIs as well.
And this is right. So you've got a capsule count at the beginning and end.
Yes, that's right. So
it's actually in some studies it's an exclusion criteria if the patients haven't been compliant with dosing.
Well, in clinical trials, you're not supposed to, exclude. That's called intention to treat analysis. So you're not supposed to exclude, even if the participant did not take the, completed the full capsule, you're supposed to include them in the final analysis. But there is another analysis called per protocol analysis. In that analysis, you can exclude people who did not complete. For example, we can put a threshold. In our study, we put 80%. For example, the people who did not complete 80% of the capsules, which they were supposed to, then we do an analysis.
Per protocol analysis.
Yeah. So, but for any clinical trial and for any regulatory or anything, you need to show people the intention to treat analysis. Per protocol analysis will be an additional supplementary analysis only. So here we actually, this is conducted in Tasmania. You can see this is the map of where the postcode of people where people came and participated in our trial. Yeah, most of the people were in Hobart, and then a few people from Launceston and then Swansea area. So we so nearly 370 participants were screened for the study, and then a lot of them became ineligible for a lot of reasons, including lack of pain and other reasons, including
up to 50-60%.
Yeah, I forgot to tell you that we did exclude people with severe osteoarthritis. It's called grade 3. We do not take usually those participants for our disease modifying treatments. For example, because in this trial we collected MRI as well, so we were expecting to see a change in the cartilage. So we want people with cartilage, because if it is too severe, you're unlikely to find any cartilage in those patients. And then if you take an MRI,
And I think Bronte, the majority of OA patients in the community are that sort of mild to moderate, right?
Yeah, that's right. So most of the people will be in that range anyway. So, and then out of the 112 people who are eligible, and then they underwent the phone screening, and they were, excluded for various reasons. If you're interested, you can see that paper. They will have, all this as mentioned in the paper, why they were all, excluded. And then 70 participants were allocated to treatment group and the placebo group. And then we did not lose many people. Only 2 people were lost to follow-up. So we analyzed, nearly 36 and 34 people in this trial. And then, so this is the I don't know. Oh yeah, probably you. This is the baseline characteristics of the participants. You can see that they were very well matched at the baseline. That means the people who were taking the treatment or the placebo, they did not have any significant difference in terms of the age. Yeah, in terms. Well, it was actually, I have to say that we used a criteria to randomize that there was no difference, so that we made sure that people in gender will be equally distributed as well. So and then
there's that implication between higher BMI, incidence of OA, incident of inflammation as well, right?
That's right. So, and then when we looked at the baseline treatment and placebo, there was no significant difference between these two groups, which is the beauty of randomized clinical trials. The idea is that all the confounders which are measured, we call it as the confounders which are measured, and the unknown confounders as well, which we haven't measured, are supposed to be equal between the placebo and treatment group. That's why people say that you need to do a randomized placebo-controlled clinical trial, double-blind clinical trial to evaluate the treatment efficacy, because even the measured and the unmeasured confounders will be equally distributed. Whereas in an observational study or in an open label trial, all the confounders won't be equally distributed.
Both variables are not distributed.
Yeah, that's right. So this is the primary outcome of the study. So you can see that the VAS pain, which is on a 0 to 100 scale, which we measured, this is the change. That means the follow-up minus baseline in the treatment group. You can see there was a reduction of 23.75 millimeter in the treatment group. And then you can see that this is the placebo effect. There was a difference of 14.64 reduction in pain. This is what is not observed in most of the clinical trials in other. So we should be expecting this, because there is a strong placebo response in osteoarthritis. So, and which makes our between group difference of 9.11 millimeter, which was statistically significant. And I'm not saying that 9.11 millimeter is a large effect, because we say that the minimum clinically important difference in osteoarthritis is considered 15 millimeter. So this is a modest effect, but compare it with the non-steroidal anti-inflammatory drugs, or even we say that the paracetamol has got an effect of only 4 millimeter. So compared to compared to paracetamol, or compared to a non-steroidal anti-inflammatory drug, which has got a similar effect of around 11 to 12, but and compared to the safety profile, I still think the turmeric seems to be can
therapeutically relevant, I think you would say.
Yeah, it's irrelevant, but it may not technically meet the minimum clinically important difference. But considering the current scenario that we do not have a really good treatment in osteoarthritis, turmeric has got a role in the treatment of osteoarthritis. Whereas we did look at the MRI defined effusion synovitis, so we looked at the volume and volume change in MRI, but we did not find a significant difference between the treatment and placebo group. So there was no difference in the structure in the MRI, which we measured. And again, this is the visual representation of the knee pain score. You can see that the reduction here, the reduction in the treatment group and reduction in the placebo group, and there was a significant difference of 9.11 millimeter here.
So to the psychoneuroimmunology of pain and the application of placebo, even if it has no therapeutic inclusion, it still has a therapeutic benefit in so many cases, right?
Yeah, that's true. And if you look at the intra-articular, placebo injections, that means in a normal saline injection studies, this will be like even more than the treatment effect of turmeric. So it will be just a placebo will be like here. So the effect is quite larger, and the best treatment will be a sham surgery. And if we just do a sham surgery in patients, that will have the best effect as well. Right? That's right. Yeah. So this is how we quantify. I'm not going to go into the details of all those 3D volume rendering and how we quantify the MRIs and all those things. But the highlight of my trials, which I do here, is that I always have some sort of structural endpoint, whether it's CT or MRI or X-ray, and we try to define some sort of structural endpoints as well.
It's your passion for orthopedics coming through.
Yeah, that's right. So and then so this is again, on top of our sequences, we used a compositional MRI as well. So we call it as the T2 values. So it's again the measure of the cartilage composition. And then we looked at the composition of cartilage, and there was no significant difference. But when we looked at the other secondary endpoints of the other pain measures, such as the WOMAC total, WOMAC pain, WOMAC functions, and WOMAC stiffness, you can see there was a significant difference. Again, the effect size here, it is modest, but it was statistically significant. And here you can see that we define responders. Responders means, people who do respond to treatment based on a criteria, based on the global evaluation and everything. We have a set criteria. So like we say that there's an absolute change of 20 or a relative change of 20%. So in that circumstance, we defined as a responder. That means this patient took medicine. Doesn't matter whether it's placebo or a treatment. They did respond to the treatment. So there is a minimum set criteria defined by the ORZ Act. And you can see that 22 people out of the 35 responded to the treatment in the treatment group. In the placebo group, whereas only 13 people responded, which means that if you are taking turmeric treatment, you're 64% more likely to respond to a treatment compared to if you're just taking a placebo, which is again highlighting the way that the treatment did work. So this is again the WOMAC pain visualized in a graphical way, and you can see that here. What is interesting here is that the clear pain difference started from week 8 to week 12, which was one of the main argument we used for our grant application, to see that we need more money to fund a clinical trial which is large enough for nearly 260-300 people, and then we want to follow them for a minimum 1 or 2 years, because it is likely to become bigger, because following the trend from here, it is likely to become, this difference is supposed to become bigger if we follow them for 24 weeks or even 48 weeks.
Assume a linear kind of progression of both curves.
That's right. Yeah. So we are assuming it. These are the arguments we put forward, but which is still not funded yet. But these were the arguments which we did put
But
forward.
I think this, if you study that graph, what it's showing is that there's some level of onset of clinical effect from day 5 for both
It's week 8.
placebo.
Oh, you mean day 5 itself?
So something's happening between
that's
0 and
right.
day 5.
Yeah,
But
yeah,
really
that's
it
right.
starts to kick off from
Yeah,
week
see.
4, week 12.
Yeah, so here day 5, you can see that
Right,
the
there we go.
wax. Yeah, there's a, the effect was there strong enough right from here itself.
Yeah.
And then the maybe it's gonna go quite higher from week 8 to week 12. This is the VAS, and then in WOMAC it's much more evident.
Yes.
Yeah.
That's right.
Yeah. And the WOMAC is a multifactorial score, yeah, whereas VAS is just basically a Likert scale of pain.
Yeah, that's right. Yeah, that's right. But it's a well-known, accepted, primary outcome for the clinical trials anyway. So the other secondary outcomes, and I'm not going to the details, but it's all we did have the physical core. These are all the recommended outcomes by the ORSI. So we did follow the standard. That's why we could publish in a good journal, because we had all the outcomes, everything required by the ORSI. ORSI is like the International Organization for Osteoarthritis. They have defined the criteria you need to be measuring in a clinical trial, and we had all those criteria. And so none of those physical function measures were significantly different between the treatment and placebo group. This is again the pain we separated into weight bearing and non-weight bearing. So the weight bearing pain will be the pain while standing, pain while walking, pain while going up and down stairs. So those three, we combined together. So in 0 to 300. So there was a moderate effect again on the pain weight bearing scale. And then non-weight bearing pain will be the pain while sitting, and then pain while at night. So those two. So because the WOMAC pain has got 5 main components. So the 5 main components I have told you, like the weight bearing and non-weight bearing. So the 5 will be the non-weight bearing will be the pain while standing, pain while going up and down stairs, and then pain while walking. And the other 2 components are non-weight bearing, which is pain while sitting, and then pain while, I think, lying down, or at night while lying down. So there was a significant effect when we separated into both weight bearing and non-weight bearing.
That's amazing.
Yeah, because the non-weight bearing pain can be more associated with the inflammatory. So we thought there might be a larger effect in the non-weight bearing, but it seems to be similar in both weight bearing and non-weight bearing. So again, on adverse events, as I mentioned, there was adverse events reported, which is any event reported. It's not adverse event. We cannot say that, because how we assess in clinical trials adverse events is anything that's gone wrong with the patient. It has to be it have to be reported. So serious adverse event is any event that is reported to the clinical trial coordinator or to the study to us, which is required hospitalization for more than 24 hours. So you can see that we had nearly 2 cases of serious adverse events, but both were in the placebo group only. So it's nothing to do with the treatment. And rest of the other adverse events reported, you can see they were well matched between the treatment and the placebo group, again indicating that there was no additional adverse events. Even in the gastrointestinal, you can see that 2, and then actually it was 4 more in the placebo group. So that means because mostly we expect some sort of gastrointestinal troubles, especially this was a Caucasian population, so I wasn't sure. I wasn't sure whether 1,000 milligrams. So now I felt I should have put it to 2,000 milligram, but but that was one of the reasons why we didn't go for a higher dose, because it was slightly different population as well. So well, if you give me another chance, I'll go up to 2,000 milligrams. That's a different story, because I want to see more effect, not my 9 millimeter difference. I want to see at least a 15 millimeter difference. Anyway, no difference between the treatment and placebo in terms of safety. So which again concludes that there is a significantly. So conclusion is that there is a improvement in knee pain, knee osteoarthritis patients with over 12 weeks, and there is a moderate effect of the treatment. And the treatment, as I mentioned, treatment did not plateau at 12 weeks, indicating the efficacy may be greater with long-term therapy.
Yeah.
I'll just go and get my charger. Is that okay?
Yeah,
just
please.
hold on a second. Yeah, thank you.
No problem. My story of my life, my computer's always going to sleep.
Always at the best times, isn't it?
Yeah, exactly. Exactly.
Really
I actually
interesting.
thought it was my power, and I turned around to see if my if the power was turned on, and it was. I was a bit worried. I'm like, what's going on?
I did the same check, don't worry. I'm not actually not plugged in. There we go.
Yeah.
Now I am.
Yeah. So back here. All right. All right. So do you have any other questions? Actually?
No, that's so comprehensive, and thank you for that. It's just as we were saying before we started recording, it just I'm really thrilled with how intelligent the study design is, and I think also that should be said for some of the other Turmacin studies as well. There's just I love it because it's not an easy thing to do well. It's very and just the number of other elements that you had to the study in terms of not just one validated osteoarthritis measure, but I think 3 in the study, plus biomarkers, plus MRI, plus ultrasound. I think it's really impressive.
Yeah, I haven't really shown you the biomarker data, but I think it's fine. I can verbally say it. I have another presentation on biomarkers, but I think, do I need to present that? But it's
No, that's fine. That's fine.
yeah. So but we did actually, in a scientist perspective, we did look at whether, I know it's probably not relevant to you, but where we looked at whether the baseline inflammatory markers predicted a treatment response. What we found was higher, if you have a higher baseline inflammatory marker, such as the IL-6 and TNF alpha, you are highly likely to be responded to treatment. So so that is what we found, because I know that probably it's not of interest to you because you're mainly looking whether the treatment reduced the inflammatory markers or not. But as a scientist perspective, in a next trial,
it's totally interesting. I think from a clinician's point of view, it's very interesting because it's almost saying, if you have patients present with higher inflammation, they're probably likely to be better responders. Is that what I'm hearing you say?
Yeah, they're highly likely to be better respond to the treatment. So that's
very relevant. That's extremely relevant.
So but it's a modest, I'm not saying it's because it's a smaller sample size study. So we published that paper actually in Phytomedicine. It's not a bad journal. It's number one, in complementary
for us
medicine.
In the herbal medicine field, that's like top.
Yeah, it's a top journal, and then we published it actually in a month, only in a month before. Yeah.
Very cool.
I'm happy to send you all those papers. So we published 2 papers from that clinical trial. So the one original paper in Annals, and then phytomedicine trial. And then one more paper related, it's a systematic review. It's nothing to do with our original trial, but I've used the data from the original trial as well.
I think the systematic review is relevant for our sort of more nerdy customers who want to look at it. Because I actually personally haven't seen a systematic review of curcumin or turmeric. So I'd be very interested to see that.
Yeah, that's a really complicated, we did a concept called meta-regression as well. So we looked at which factors predicted treatment response. So what we found that, you're highly likely to, your effect of turmeric treatment goes down with higher BMI. So if you have, as the BMI of the patient goes up, so your response is lower as well. So that's it's called a meta. It's actually not from original data. So it's from the regressing our coefficients from the different studies into the effect. So I can send you all those 3 papers together, and then you can just go through all those things. Yeah.
Very cool. So question, do you do your own stats or do you have a statistician on staff?
Yeah, I do, because I've got a PhD as well. So I did I had to do all
research yourself.
Yeah, so I do all those statistics. First of all, I do it, I learn it from a statistician, and then I do it myself.
Then you do it yourself.
But then for this clinical trial, I'm the PI, so my PhD students, I taught my PhD student, he had to do it. So everybody had to learn it. So I didn't do it physically myself.
Discipline in and of itself, and I think I have had, when running clinical research before, not as the PI, but just as more like the sponsor, I've had to engage the services of a statistician just because I didn't trust the analysis I was getting coming out of the study itself.
So we have a statistician in the team. For example, one of the authors is a statistician. So all our analysis is vetted by him. So for example, so we do it, but we always show him this is what we're doing and this is it, and whether it's correct or not. So he will say yes, but he will not do it for us, because it's like this is a PhD training process as well. So I was a PhD steering, and then a PhD student, and then I become a guide, and my student doing. So it's all about training process as well.
Amazing. Thank you. Dr. Benny, what's next for you and your collaborators in research? What are you next looking at researching?
Yeah, so actually currently we are actually finalizing fucoden, so we are going to do a clinical trial in fucoden. There is
on the Tasmanian material?
Yeah, it's
wonderful.
that this company, right?
Yeah.
And then now I'm actually, I'm in discussions for one of the, cannabis extracts for the treatment of OA. Yeah, because that the cannabis extracts seem to be recently been TGA, so Schedule 3 drug, now. Right. So I'm trying to get into that space, and then, if I can try it for either rheumatoid or osteoarthritis. Yeah.
Fantastic.
So yeah, clinical trial ideas. If you want any help or anything, just let me know. Yeah. I'm happy to conduct the trial anytime, anywhere with a reasonable, scientific, a question. If we can
I mean, honestly, I'm so excited that you're based in Australia, and two, you've got this very, very practical, pragmatic approach to clinical research. It's great.
That's right. Yeah, because some of the things which I want to do, for example, the colchicine trial, I'm still trusting the drug colchicine, but it's unlikely. No one is going to fund that trial. The problem is that, even I've been in touch with Zydus and all those things. So nobody, because it's out of patent and no one is commercially interested. So a lot of the scientific questions which I have, because I wanted to select people with, I don't know whether with evidence of crystallization in the cartilage, and select those patients and give them the colchicine. I still believe it's going to be working, but no one wants to fund those trials. But yeah, so I go with the flow.
Well, so listen, everyone, thank you so much for your time. Really, really appreciate it. I know it's into your family time, so thank you very much for taking the time.
Thank you.
Yeah,
Thank you.
That was a wonderful discussion to be a part of and hear Gabriel and Betty going back and forth. So that's fantastic. And I have to say, we're trying actually to, for Gabriel and Bronte's point of view, we're trying to get Dr. Zipak to come at the May CMA Innovation to talk about the evolution of studies, and listening to Dr. Antony, Betty, listening to you talk about the study. That is a fine example of how studies are now done, or how they can be done now, and how much better they are, or how much better they can be when they're done properly and by somebody with the passion to do it, to show the efficacy of these natural products that we have. So that was wonderful. Thank you.
But I think, Benny, you're setting the standard in Australia, honestly, having met and worked with many of the other researchers and clinicians looking at natural compounds, irrespective of therapeutic outcome.
So don't think that we are centered just to natural therapies.
No.
My current trials which we are doing is the intra-articular stem cell injection trial. We are partnering with the company Turmerosaccharides, so they're providing us the stem cells. So we're right now doing a very big trial of 400 people. Wow. So they undergo 2-year-long trial. So intra-articular stem cell injections. Currently, we are doing a Dasarin clinical trial. I don't know whether you've heard drugs. It's again, it's a drug. And then we're doing methotrexate clinical trials. So there's a lot of trials going on. But because what I'm focusing, and then the stem cell trials is again, I'm part of, but
super exciting field.
Yeah. So
from a rheumatology, osteoarthritis point of view, super
that's
exciting.
Right. Yeah, that's right. So a lot of trials get involved. Yeah.
And you said you're on the BBC tomorrow.
No, actually they recorded it long back.
Oh, it's got done already.
Yeah.
Okay.
So it's like a radio to Dr. Mosby's or something. Mosby's.
Oh, yeah.
Something. Yeah. So it's like a health benefits of turmeric or something. I don't know how it's gonna go. I'll forward it to you if it's good.
Yeah, send it through, please. That'd be great.
I haven't heard it.
Feel free to share the papers we discussed as well in the conversation. We'll absolutely make that part of how we bring this to the audience.
That's right. Yeah. Thank you.